Okay, welcome to our discussion of cell division called mitosis. We'll save meiosis for another set of notes. Please make sure that you have the note packet so that you can follow along quickly. This set of notes is fairly long. Okay, there are two types of cell division. The cell division that we're going to focus on today is called mitosis. Mitosis is cell division where the two new cells are identical to the original cell. Your body goes through mitosis when it needs to repair an injury, when it needs to make new white blood cells for fighting disease, when it needs to replace skin cells that are being shed, or when an organism needs to grow, for example. That's mitosis, where the cell simply copies its DNA and then divides in two. Meiosis, on the other hand, is cell division we're going to learn about later, where the cell divides twice and it ends up with cells that have half the DNA, and those are your gametes. The only time that happens is when your body is making sperm or egg, or if a plant is making pollen, for example. Okay, this diagram here shows the life cycle of a cell, and if you see just after the orange pie piece or gold pie piece, you see the G1 phase. That's the phase where the cell is starting to grow. After the cell has just been born, so to speak, it grows during that time. We call that the G1 phase. During the S phase, following that, the DNA is copied. Remember, it makes sense that the DNA has to be copied if a cell is going to divide, because if the DNA doesn't get copied, then the cell divides and you would have half the DNA, and then you'd have half again the next time it divided. So the DNA has to be copied, and that happens during the S phase. Then it's followed by the G2 phase, which is another growth phase, and it prepares for cell division. During the M phase, that's mitosis. M stands for mitosis, and that's when the DNA is being copied. Cytokinesis is when the cell membrane pinches in, and then you have two new cells that start out in the G1 phase again. So it's a cycle. The majority of the cell's life is spent in the stage called interphase, which is that entirely blue stage. Very, very tiny portion of its life is actually spent in the mitosis phase, not unlike a human female. The majority of her life, she's not pregnant. Just a tiny part of her life, she might be pregnant. Just as an FYI, some cells can enter a phase called the G0 phase if they were no longer divide. Example is human nervous system cells. If you get into a car accident and sever your spinal column, it doesn't repair itself because those cells don't grow and divide again. They've entered that G0 phase. Now, the cool part about a cell is that it's full of different on and off switches that control how the cell divides, and that's what we're going to talk about next. At critical points, further cell progress depends on a central set of switches regulated by cell feedback. If you see at the G1 point, there's a checkpoint. The cell literally looks at itself and makes sure that it's growing correctly. If it's not, it stops cell division and goes back and tries to make corrections. At G2, the DNA replication is assessed. Did it copy its DNA properly? Are there mutations? Is it safe to continue on? If it's not safe to continue on, the cell actually stops itself from dividing, makes repairs, and then checks to see if it can continue. And then again, in the M stage, there's a checkpoint where mitosis is assessed. Did the cell, did the nucleus reform correctly, et cetera, et cetera. If those checkpoints fail and cell division continues when it shouldn't, there's the possibility of cells dividing when they shouldn't, and that can lead to something like cancer. Okay, so mitosis is the name of that part of the cell cycle where the cell is dividing, and mitosis is divided into really four stages. The first stage, called interphase, remember that's the blue stage in that diagram we just saw. That's when the cell is growing, it's copying its DNA, it's doing what the cell normally does. So really, mitosis doesn't start until prophase, and then it's a gradual process. It moves from prophase, then to metaphase, then to anaphase, then to telophase. You should, as you look at those diagrams, you should see how the nucleus is changing, and it often looks just like this, and so we can identify the name of the stage based on what it looks like. To remember the stages of mitosis, we say a stupid phrase. We say IPMAT, and that's the first letters of each stage. So I interphase, P prophase, M metaphase, A anaphase, and T telophase. I know it sounds stupid, and I'm sorry for anybody named Matt, But whenever I'm looking through a microscope at these, I'm always saying to myself, I P-MET, and that helps me keep track of which stage comes first, second, third, fourth, fifth. Okay, at this point we'll go into a little bit of detail on each of the stages. As I said already, interphase is the normal growth and normal cell activity stage. DNA is still wound up in that chromatin form. It's messy, it looks like spaghetti. We don't see those chromosomes yet. That's the interphase stage. The next stage, the first stage of mitosis, is called prophase. Here the nucleus disappears. So notice that you've got bivalent chromosomes. They look like X's now. The DNA looks like letter X's instead of that spaghetti form. So it says the chromatin thickens to reveal chromosomes. The centrioles is that little circle and box. The centrioles grow something called spindle fibers, and those are the little fingers in this picture that are reaching across toward the chromosomes. They are eventually going to grab the chromosomes and push the chromosomes into the middle. So during prophase, the nucleus disappears, chromosomes up here, and the centrioles move to opposite sides and start growing the spindle fibers that are going to line those chromosomes up in the middle in the next stage. Okay, this next stage after prophase is called metaphase, and I emphasize the M in metaphase. You might want to write the word middle next to the word metaphase. During metaphase, the chromosomes are pushed to the middle of the cell by the spindle fibers. They line up in the middle or along the equator of the cell. The next stage is called anaphase, and next to the word anaphase, you might want to write the word apart. A for anaphase, A for apart. The chromosomes are pulled apart. It says spindle fibers pull the chromatids to opposite poles. This means an identical set of chromatids moves to each side. Remember that the left side of a bivalent chromosome, that letter X, the left side is identical to the right side. So if they're being pulled apart, identical DNA is moving to the left side and identical DNA is moving to the right side. Okay, in the final stage of mitosis, the final stage is called telophase, and the way I help students remember that is the letter T. Telophase has two cells. The nuclear membrane reforms, so notice we've got a nucleus again, and the DNA doesn't look like chromosomes. It doesn't look like those X's anymore. It returns back to this form that's called chromatin, where it sort of looks like spaghetti. Now, technically, mitosis is just the division of the nucleus. So there is another stage called cytokinesis, which is where the cell membrane itself begins to divide. I always joke that this picture looks like a baby's butt, but that's showing an animal cell dividing. Now, it is an animal cell, and we have to be clear because a plant cell looks different. Because a plant cell has a cell wall around it, it can't smoosh in the way the cytoplasm smooshes in cytokinesis for an animal cell. So instead, the cell wall, it says here, the cell wall prevents cleavage. So instead, a new cell wall called a cell plate is formed. So notice there's those two dark green blobs in the middle of the picture, and then there's a faint wall forming in between the two of them. That's called a cell plate, and that's how a plant cell divides. Okay, now that's it for the mitosis and how mitosis works. But it turns out that mitosis is really, really important in why cells die and why we age. There are several theories about why we age and why cells die. The first theory, it's not really a theory, it's pretty well proven at this point. Apoptosis or apoptosis, people pronounce it differently. That's called programmed cell death. What that means is that within our DNA, our cells are told that they can only divide about 50 times. It's called the Hayflick limit, named after the scientist who figured out how many times cells can divide. The fascinating part is that cancer cells can continue to divide past that, and that's what leads to tumors. When we have more than 50 divisions, we start getting this mass of cells, and a mass of cells is a tumor. So the question is, do cancer cells hold some sort of a secret to increased lifespan? And we think that the answer is yes. Let me explain something really quickly. There was a patient in the 1950s whose name was Henrietta Lacks, that was her name. Some of you may have heard of her. Anyway, she was a cancer patient. It's a fascinating story. I don't have time to go into it. But she died of cancer, cervical cancer, I believe, back in the 1950s. But they grew some of her cells before she died in a laboratory. Now, these are cancer cells, and they don't experience the Hayflick limit. So these scientists that were studying her cancer cells, they were able to get them to grow in a petri dish, and they kept growing and growing. So they transferred some of the cells to a new petri dish. and then to a new petri dish, and they kept growing and growing and growing. Her cells today are still alive. Countless divisions have happened. They've been transferred to, her cells have been transferred to laboratories all over the world, and science, cancer doctors all over the world have been studying her cells. So it's fascinating that although this woman passed away of cancer in the 1950s, her cancer cells continue to live on, and they are not obeying the Hayflick limit. So one of the questions about why we age, one of the theories is that we build up over time random cell damage from something called free radicals, but also DNA mutations or the gradual buildup of the body's own waste poisons. What are free radicals? I'm not going to go into the details now, but just be aware that free radicals are extremely unstable molecules with one unpaired electron, and they go tearing through our bodies. And we get these unpaired electrons from foods that we eat from the process of making energy. And so they travel through our bodies at enormous speed, and if they hit our DNA, they can cause a mutation. They steal electrons from other molecules, and they can destroy a cell, which can lead to cancer and heart disease, and we think other diseases as well. Well, the secret to preventing damage from free radicals is eating foods that are high in antioxidants. Antioxidants are kind of like catcher's mitts. They sort of stop, like they catch these electrons, and they prevent the electrons from damaging the cell. They tend to be foods that are high in vitamins C and E. So diets, people that have diets that are high in vitamin C and E are less likely to develop cancer and apparently age more slowly. Okay, the last thing we'll talk about why we age has to do with something that I think is absolutely fascinating. It turns out that on the tips of all of our chromosomes, we have these sections of repeating DNA. It's these letters that repeat and repeat and repeat, and they're on the tips of our chromosomes. I've heard scientists describe them like the caps on the end of your shoelaces. If the caps on the end of your shoelaces fall apart, or crack away or whatever, the shoelace starts to unravel. Well, it appears that these telomeres, these tips on the end of our chromosomes, protect our chromosomes. And every time that the cell divides, those tips get a little bit shorter. So imagine every time you tie your shoes that the tips on your shoelaces got a little bit shorter and a little bit shorter. Pretty soon they become so short, the tips on these chromosomes become so short that the DNA might actually become damaged. The DNA in the chromosome might actually become damaged. So the cell says, we got to stop dividing. Well, that's the Hayflick limit. That's that 50 divisions. So you hit the Hayflick limit when your telomeres have shrunk down to a point where they can no longer protect your chromosomes. So that's apparently when our cells stop dividing. We reach that apoptosis or apoptosis when our cells don't divide anymore. It's also called senescence. And that's what aging is. The part that's absolutely fascinating is that telomeres don't get shorter in cancer cells. So remember we said that ladies' cancer cells are still alive today. The reason those cells are shorter is that the cancer has figured out a way to protect the telomeres. So now there are scientists who are actually looking into an enzyme that's called telomerase that protects the tips of the chromosomes. And they found that if they give telomerase, if they give this enzyme to organisms, like they've given it to flatworms, they've given it to mice, they greatly, greatly, greatly increase the lifespan of the organism by 100 times. I don't know, I made that number up. I don't know the exact amount, but it's just crazy how they can lengthen the lifespan. But there's always one side effect. Just imagine it, see if you can figure it out. If the telomeres stay long and the cell can divide forever, what's the side effect? Almost always. Well, sure enough, cancer. So organisms whose telomeres are protected develop cancer. So it appears that allowing our telomeres to shorten protects us from cancer. Kind of a catch-22. We don't get to live forever because we can't have cancer. All right, we're finished with this lecture. Whew, you survived. This is just a final slide to see if you can name the phase of mitosis or interphase. So look around. What you're looking for is which cell looks like it's in interphase, which cell looks like prophase, metaphase, anaphase, and telophase. I think telophase is the easiest one. Telophase is two cells. So which one looks like it's two cells? If you said the one in the upper left-hand corner, you'd be correct. So that one's telophase. I think anaphase is the next one. Anaphase apart. Where do you see the DNA pulling apart? It looks like fingers pointing at each other. If you said it's the second cell on the bottom, you would be right. The next one would be metaphase when they're in the middle. That's the bottom left-hand corner. And then interphase. In interphase, you can often see a nucleolus, a little dot in the nucleus. So that's the third cell on the bottom. And then finally, prophase. I didn't mention this earlier, but prophase, I'm going to emphasize the P, often looks like polka dots. Prophase polka dots under the microscope. If you look at that far right-hand corner one, you notice little polka dots of light pushing through the nucleus, and that's because the DNA is winding up to form chromosomes and it doesn't take up as much space, so light passes through. So interphase is just to the left of it. The nucleus looks solid with a polka dot in it. I'm sorry, with one nucleolus in it. A polka dot was a misleading thing to say. And then the far right-hand cell has little polka dots of light passing through, and that's what allows us to know that that is prophase.